Okay, good morning. We're actually a little ahead of where we were last semester, so either we can slow down or we can keep going. So we may get into a little bit of PowerPoint number lecture 5, which you don't have. You'll have it by the end today, or by the end of the day. We may or may not get that far, so I'm going to try to stay a little bit further ahead in getting you those PowerPoints and outlines. But we will be on outline number 4 for more than one lecture because it takes that amount of time. Where we are now, we've sort of finished the basic review. This is the type of information you should have gotten out of genetics class and also introductory biology about how information is unrolled from DNA and then rolled out into a program that determines what you and I end up looking like and functioning. So basically, information is stored, it's encoded in chromosomes, and it's encoded in a really complicated way, and so we need to see how that information is stored. But there's actually a level of control above the genetic level, which is called epigenetics, and we'll get into that, but it's how the DNA is folded and unfolded so that it can be read. And then, of course, the DNA is transcribed into RNA. Some of the RNA is things like messenger RNA or transfer RNA. Others are actually different types of information, such as microRNA and small interfering RNA that we'll talk about a little bit later. But RNA is much more complicated in its folding pattern. And the simplest folding pattern is DNA. It pretty much likes to be a double helix. You can wind it around proteins, but just by itself, it doesn't fold into very complicated structures. So it's a really bad information carrier. It's very good to store it, but it's really bad at carrying lots of information in a very small place. RNA is much better because it can fold into secondary structures. Think about the stem loop structure of transfer RNA. It base pairs to itself. It's got open loops, and so there's actually secondary structure and actually tertiary surfaces that can carry information and can be recognized by proteins. And then the ultimate, of course, is protein. It is the most complicated folded macromolecule in the cell. It carries lots of information in a very small space. So that's why we pretty much translate everything functional into protein. Okay, and so let's go and review these control points, and I'll show you where we're going to get started, which basically is at the nuclear level. So we're in chapter 4 now, and it's focused on DNA, chromatin, and beyond. Okay, so we want to identify the components of the nucleus and what is chromatin, by the way. And so we need to understand that information before we can look at how we take it apart and open it up so that it can code for proteins, messenger RNA proteins. So we want to look at the organization of a typical gene. That should be reviewed for you, but if it isn't, this is a good chapter to go over that process. We're going to explain how so much DNA can be packaged into such a small space. Basically, you're packing all your DNA into a nucleus is like taking, your book says this, a 24-mile long piece of thread and putting it inside of a tennis ball. That's the same packing problem. So there's something really going on about how complicated that the DNA can be looped around. DNA doesn't fold very much by itself, but it can sure be wrapped around and controlled by proteins. Okay, and then we'll look at how chromatin-Rivalli complexes and histone chaperones alter gene expression, because you got to unfold DNA before you can read it. Okay, so here's the basic. We're going to be on this figure actually for quite some time because we actually started down here talking about proteins. This control point is where you take a protein and several things determine whether it's active or inactive. And we used as our model enzymes. So you now know that proteins can be activated or inactivated by covalent modifications like phosphorylation. They can be inactivated or activated by cleaving them. So that's where you are right there. And so there's all this control point in between where we have to step back and take a different look. And we're going to actually start up here in the nucleus, and we're going to actually begin with epigenetic control. How is the DNA folded, stored, and then how is it unfolded so that the transcriptional machinery has access to it? If this DNA is not unpackaged, transcription doesn't take place. So we can't even get to this level of control. The next chapter that we'll look at is actually transcriptional control. You should have had that ad nauseum in genetics, but we will take a slightly different look at it again. And then the RNA transcript goes through a whole series of processes that turn it into protein, right? And then, of course, you come back to where we started with enzymes. The protein level can be independently controlled. But we have a lot to do up here, and the way we start is looking at the structure of the nucleus, which you should have had since second grade. You know, learned to read and you saw the nucleus. And so this is pretty much what it looks like in electron micrograph. This is the nuclear envelope, the boundary of the nucleus. There is a couple of different types of DNA. You have heterochromatin that's highly packaged, and so that's what defines heterochromatin, is it's all tightly packaged so that it's not usable, it's turned off. In the middle here you have what's called euchromatin, and that has the ability to be transcribed. Okay, that's basically the difference. That doesn't mean it is being transcribed, but it is fairly unpacked, and we can look at how you get to that point so that we can turn on genes. Okay? The other little obvious piece of architecture is the nucleolus. What's going on in here? Why this big stain?
It's heavily packed with ribosomes.
Yeah, that's where all the genes that code for ribosomal RNA are located. And this really is a measure. How big this thing is in relationship to the diameter of the nucleus is a measure of how active that cell is. So if this cell is cranking out proteins, this thing is really big and really active, because that's where RNA, ribosomal RNA, is a component of the ribosome, and those ribosomal subunits are constructed inside the nucleus, and then they're ejected out through a nuclear pore. Okay, so now let's look at the cartoon. We've seen the real thing. And so just a couple things to point out that are often missed. So the nuclear envelope is the border between the nucleus and the cytoplasm. It actually has three components. There's an inner and an outer nuclear membrane, and then there's this space that's called the perinuclear space. That's just the same thing as the lumen of the ER. The ER is the nuclear envelope smushed into the nucleus. That's what it is. Okay, so this portion of the ER could be loaded with ribosomes, and any kind of protein that gets into this lumen actually communicates and goes into the perinuclear space. So they are one and the same, ER and the nuclear envelope. Okay, then you have all these little holes here. These are nuclear pores. There's anywhere from 3 to 4,000. All the traffic that's big that goes in and out, like messenger RNA and all types of ribosomal subunits and big proteins, anything that's above a certain diameter must go through this nuclear pore. Very few things can diffuse across the nuclear envelope. It would have to be very lipophilic to be able to do that. So most of the traffic goes right into here, and the more nuclear pores you have, the more active that nucleus is. Okay. All right. So let's go into a little bit further and looking at the nuclear envelope. And there it is, of course. This is a little bit more complicated or specific diagram, and I show this not for you to memorize all these components. But let's imagine, so there's the ER, this is the rough ER, and this would be smooth ER where there's no ribosomes, and then it turns into the nuclear envelope, the outer and inner nuclear membranes, okay, perinuclear space, which is communicating with the ER lumen. So this ER lumen is a oxidizing environment where disulfide bridges are formed, as we learned. You have this huge machine that's called the nuclear pore complex, and which actually ties pieces of the nuclear envelope together. And then you have these little railroad tracks, this green stuff. Now this is the nuclear lamina. It is made out of a cytoskeletal element known as intermediate filaments. Okay. And if you were going to try to break the nucleus apart, and I mean during cell function, when is it that the nuclear envelope is broken apart in a normal cell cycle? let's remind ourselves of the cell cycle, first of all. So it's a circle, right? There's mitosis, you go to G1, you go to S where the DNA is duplicated, you go to G2, then you go back to mitosis. Okay. Where in that cell cycle do you actually need to dissolve the nuclear envelope? Mitosis. In what phase?
Interphase.
Which, you remember the 4 phases? Pro, meta, ana, telophase. Where does it go away? Prophase, late pro-metaphase, actually. Okay. And so we actually have to break this apart. So which structure could you dissolve and then get rid of the whole nuclear envelope? So what's happened here is all this, look at how things are attached. The chromatin is attached to the nuclear lamina. The nuclear lamina is bound to proteins which are attached to the nuclear envelope. The nuclear envelope is held together by the nuclear pore complex, which is in turn attached to the nuclear lamina. So if there's one thing that you could break apart that would tear the entire nuclear envelope apart, what would it be? Just taking a guess. What's the most labile piece? The lamina. The lamina is actually the piece that your cell decides to phosphorylate during cell division. And that phosphorylation of this nuclear lamina makes it fall apart, and the whole nuclear envelope goes away. The ER turns into little vesicles, and then that allows the microtubules, the spindle fibers, to come in and bind to the chromatin after it's been packaged very heavily into chromosomes. Okay, so the key thing that's holding this apart is a very strong structural element. And as we go into cytoskeleton here in about 2 months, it's one of the last topics that we talk about, the intermediate filaments are designed to do one thing. They're structural elements. They hold things together and don't let go unless they're told to let go, like phosphorylated. Okay, so nuclear lamina is important. The nuclear lamina also, note, has a hand onto chromatin. Okay? Often that chromatin that's bound to the nuclear lamina is heterochromatin because it's holding onto it at a place where it's not going to transcribe the DNA, so it's a sort of a structural hand grip onto the chromatin. That helps organize the chromosomes within the nucleus. As you'll see towards the end of this lecture, at the beginning of the next one, the chromosomes are very organized. They're not just floating around willy-nilly in there. The nucleoplasm is a very complicated and organized place. There's a place where transcription is going on. Here's pole 2, reading DNA, and there's the spliceosome that's splicing the exons together. And there's all kinds of information. There's transcription factors that actually bind to the nuclear lamina, and they're floating around. So, and here's a strip. This is the chromosome right there. This is chromatin is what? It's protein plus DNA. And as you'll see, there's 2 types of protein forms chromatin. Okay, so again, this is a very complicated place, so we have to have a way of organizing it. All right, so this is a good way of organizing it. The only time you actually see DNA in this shape is during mitosis. It does not look like this during interphase. It's not as compact. The reason it needs to be compact during mitosis is that you're moving the information. And what happens if you leave a piece behind? You get a retarded cell, right? Or the cell dies, right? Or you get cancer. All those things could be true. This is a technique called chromosomal painting, in which each chromosome is individually painted a different color so you can distinguish them. Of course, when they spill out of the cell, they're all mixed up. And so somebody has gone to the trouble of lining all of the 22 autosomal and the 2 sex chromosomes. This is obviously male, XY, up there so you can see them. So 22 pairs of somatic chromosomes, 1 pair of X chromosomes, of sex chromosomes. Okay, so this is the only time you'll see it this organized is during mitosis. Because when it's packed that tightly, you cannot read it. You cannot read the DNA. You cannot transcribe it. You have to unpack it. All right, so we've identified some of the components of chromatin structure, and let's go down a little bit further in, this is chromosome number 22, and notice 22 is really tiny. This is this very small, simple chromosome, but it's still got lots of information. When you display all the chromosomes like this, that's called a karyotype. When would you like to do this? When do people actually look at this clinically? Yeah.
If they're looking for an aneuploidy.
Aneuploidy, that happens in what disease? Often.
Down
Trisomy 21.
Well, Down syndrome, you know, developmental diseases, but Dr. Kogos that was here, cancer. A lot of, you'll look at the cancer karyotype, and you'll have, you know, a piece of, particularly in the disease he was studying, 16 has been inverted and it's attached to somewhere else. That really messes up the control over whatever is on that chromosome. And so a karyotype gives you a bird's eye view of how abnormal that person's tumor cell is. Okay? The more messed up, the more cancer-like it's going to be. All right, so that's useful. But let's break down this thing. This is big. Okay, so this is highly packed. And if we just take a 10% little chunk of this and spread it out, we'll get about 40 genes along there. And if we take about 1% of this, you know, it's another 10% of that, but it's 1% of this, you've got about 4 chromosomes, 4 genes, okay? And notice that the idea here is that these genes are spread apart. There's lots of space in here. And we used to call this junk DNA, but now we know it's not junk. It's actually information about where proteins are going to sit to transcribe or to modify these various genes. So information is packed in here, and we're learning to decode that information. Okay, and if you take that one actual protein coding gene, the classic pieces is there's some regulatory, usually upstream from the start site, which would be here. Okay, so this would be +1. The numbers going this way are negative numbers, right? And numbers going this way are positive numbers. And since a molecular biologist did this, then there's no zero. They suck at mathematics, so there's no zero nucleotide. It's either +1 or -1 going 2 different directions. Okay, so the protein coding gene, of course, the actual information is stored where? The part that you're going to turn into messenger RNA.
Exons.
Exons. See, this other stuff, it's not junk, but it's not going to be turned into messenger RNA. So the actual exons are actually really small. They're about coding for about 150 base pairs. And this could be 2, 3, 4, 5,000 base pairs. So all the information is spread out. And this gives the regulatory machinery chances to alternatively splice this. And so it allows the control over this to be exquisite. We can make more than one mRNA from this transcript, depending on how we splice together the exons. Okay? And then finally, when you splice all this together, the messenger RNA, again, there's only a piece of that is the protein coding region. You've got a 5' untranslated and a 3' untranslated region that also has different functions, but it is not to be turned into a protein sequence. All right, so fairly complicated process to get to this part right here. So we actually need to back up and understand how you actually go from basically here all the way to here and then to here. Okay? And that's a packaging problem. Okay? So the packaging of DNA, I already pointed this out, but just briefly, DNA is packaged different ways during the cell cycle. So in the interphase, in G1 is when it's going to be the most unpacked, right? It's open for transcription. Parts of it will still be heterochromatin, but most of it's going to be euchromatin, and you can turn on the correct genes, right? Okay, and so then S phase is when you're going to duplicate each of the chromosomes, and then mitosis is when you take that duplicated chromosome and heavily package it so you can move it. And so that's when it needs to be the most heavily packaged. Least packaged, most heavily packaged. Okay. All right, so this is what we associate. This is a scanning electron micrograph of a fully mature mitotic chromosome. And it looks very simple, but then, of course, this is what it's actually holding. This is all the DNA that's been unraveled and allowed to spew out of that chromosome. So we really have to ask the question, how do you go in here and get one of these threads and organize it into a way that makes this thing appear? Okay. And we start at a fairly basic level. This is called the beads on a string level of DNA. Okay? And so what you have is, a series of proteins, little structures called nucleosomes, around which the DNA is wound, about 2.5 times. And then there's a spacer, and then there's another nucleosome. So one way of folding the DNA is to take a nucleosome and wrap DNA around it. So we need to understand that process. Because when you want to go up to the next level, which is called the 30 nanometer solenoid, you have to take this structure and pack it together. And this is still a long way from being a mitotic chromosome. But we're getting there. Okay, so you never have naked DNA floating around very much at one time. Instead, you wrap it around what's called a nucleosome. This is the core particle of the nucleosome. And you can digest the DNA, and you find out that basically it's what's wrapped around the nucleosome and this little linker together. That's a total of 200 nucleotides. It's about 100. I think it's 147 to go around these. Wrap around the nucleosome a couple of times. And then the linker is about 50. So it's 50 and about 150 wrapped around there. Okay? And so if you were to dissolve all that, you could release the nucleosome with the DNA wrapped around it and break it apart. And if you did break apart that core nucleosome particle, what you get are 4 different proteins. 4 different proteins. And we call those proteins histones. There's histone 2A, histone 2B, histone 3, and histone 4. And there's two copies of each one of those 4 different proteins. So what makes up the nucleosome core? It's an octamer. How does that octamer come together? Okay, well, let's look at that question. So histones are all derived, they're all derived from one another. Okay, and there's no computers allowed. Sorry, check out. No computers in the class.
I'm taking my notes on it.
That's fine. I don't allow that. You have to take it on paper.
So I should not take notes for the rest of the time here?
You should take notes on paper, but you're distracting people behind you. If you read the syllabus, you'd see that that's the case. And you can come talk to me about that later, but... Okay, so there are these 4 different histones are all related to one another. The key structure that they have is called the histone fold. Okay, and that is the motif. It has, it's got a helix, a small loop, a fairly long central helix, and then another small loop and the C-terminus. So that's the histone fold. And H2A and H2B fold together. And the way they fold together is by forming this structure here. When H2A comes together and H2B, that's called the histone handshake. And H3 and H4 form a histone handshake. So you have these 2 guys shaking hands and these 2 guys shaking hands to make dimers. And then we're going to construct from those dimers the rest of the core particle. Okay, so what happens first is H3 and H4 do the histone handshake. They can do that because they have the motif of a histone fold. Okay. You get a histone dimer, H3, H4 dimer, and 2 of those come together to make the internal structure of the core particle, which is a H3, H4 tetramer. So it's 2 handshaking H3, H4 stuck together. That's in the middle. Okay. And so you can think of that in this little, this is like the top of a centrifuge tube. This is the core particle, and it's formed by this, it's formed by this H3, H4 tetramer. And that's what the DNA wraps around. DNA is going to wrap around that a couple of times. Okay. Now, to complete this core particle, you need to have H2A and H2B handshake and form 1 dimer, and then H2A and H2B form a second dimer. Where do they go? Well, the first dimer goes, if you have a coin in your pocket, like a quarter, you can do this. So one of those H2A, H2B dimers goes on top, and another goes on bottom. So that's how you put this octamer together. to construct the nucleosome core. Okay? And that allows you to wrap up the DNA. And notice something interesting about this. If we go back to the sequence of that histone gene, what you have are the N-terminus has some really long extended ends, the N-terminus. And those N-termini are actually very flexible, and they project away from the outside of that core. So you can see them flailing around. And that's going to be important for two reasons. One is we're going to write a histone code onto that protein by modifying it chemically, covalently. So that's where part of the histone code is written. The other thing that that N-terminus does is it latches on when the nucleosome starts to pack together. These N-termini grab on to an adjacent nucleosome, so it helps hold the packaging together. So it's got a structural and a regulatory function. And so let's go into the structure of that. Okay, so the nucleosome in and of itself is very dynamic. Don't ever think of this as being a static thing and then you just sort of put it in a box somewhere and it just sits there. Instead, what's happening is the DNA is wrapping around and unwrapping on a very short time scale. So it may be stable for 250 milliseconds, and then it will unwrap for 10 to 50 seconds, and then it re-wraps. Why is it doing that? Why is it vibrating between these two different states? Well, look at what's going to happen. You're going to have a protein. Let's say you want to turn on a gene. The protein has to have access to the DNA to read it, correct? So a transcription factor cannot read this if it's stuck on top of, wound around a nucleosome. That's the key point. You have to expose it briefly, and this is always vibrating back and forth to give it the chance to be able to recruit a protein. That particular cell has to be able to make that specific DNA binding protein, and then that's the first thing. You have to have the protein made. It needs to be in the nucleus, and this needs to be vibrating in conformation that shows or reveals a DNA element to which it can bind. So this is very, it's allowing it to be expressed, but it doesn't stay open forever. It's just going to give a brief glimpse and it's going to rewrap. Okay. But we can control this packing very exquisitely depending on how we write the histone code. Okay, so there are a series of proteins that their job is either to pack the DNA more tightly around the nucleosome. These are called chromatin remodeling complexes because they change the shape of the protein to which DNA binds. So it can, in this case, what it's doing is that it's actually, these are all ATP aces. They split ATP and use that energy to actually move the nucleosome and unwind it. So it's actually grabbing onto the protein and unwinding it and exposing. It's just like a little fishing reel that's coming out and exposing this little piece. Okay? So this briefly is going to expose that, and if there's a transcription factor that is that can bind to that little piece of DNA, which would be called a DNA response element, then you've got the start of gene transcription. Okay? Alternatively, you have some chromatin remodeling complexes that make them go tighter. So some remodeling complexes make it looser, some of them make it tighter. So here's the rule. If you make the packing tighter, you decrease transcription. If you increase the release of the DNA, you make it looser, you unwind it from the nucleosome, you make it easier to transcribe a gene. Okay. Now, so these remodeling complexes can sometimes they actually eject some of the histones from the core particle, okay, and replace those with a different set of histones. As you'll see here in a few minutes, there's actually different isoforms of each one of those, H3, H4, H2A, H2B. So there are isoforms that can be incorporated that changes the function of that nucleosome. Okay. And you can actually eject an entire nucleosome freeing the DNA momentarily, and then it quickly picks up another nucleosome and rewraps. In the meantime, this can be transcribed. And it's just this very small little region of DNA. Okay? So we have exquisite control over the relationship of packing of DNA to protein. Now, when you release these proteins, they don't just go flying off. They usually bind to histone chaperones that keep them folded properly. Because without its binding partners, that's gonna, if they unfolded, that would be a bad idea. So they're usually caught up by histone chaperones that will keep them folded, and then they can be released in their proper folded state to reassemble into a nucleosome. Okay. Any questions so far? This part's all, yes.
Is it the case that all nucleosomes are the same? They don't require any special areas to be in?
They differ from each other if you insert some of the isoforms that we're going to look at. Is that what you mean? You can get different versions. Say H3 has there's several different isoforms of H3. And so if you insert one of those, that makes that nucleosome different from a regular nucleosome.
And how does that impact the...
I'm going to show you. Okay. Hold that thought. Anybody else? Okay. Now, if we want to go from the beads on a string, which is the level where we were, up to a 30 nanometer solenoid, we actually need two things. Number one, the N-termini need to grab onto each other so that the N-termini of this histone core particle will actually grab out and reach over and grab onto this nucleosome. So that's where the N-termini come into being a structural element in packing. The next thing you need, though, is, so we want to go from this to that. We need a different histone that is not part of the core particle. We need an H1 histone. And what it does is it sort of fits right into this little nook in the linker part of the DNA that's between the two nucleosomes. And that helps it package into this 30 nanometer solenoid right there. Okay, so to get this, you need the H1 histone outside of the core particle, and then you can form that. Okay? So now we've been able to pack it. If you package it into a 30 nanometer solenoid, the DNA is turned off, but it could quickly turn back on. So it's not heterochromatin at that point. When you go to heterochromatin, that's when you turn the gene's actually silenced, and you can get what's called epigenetic effects. Okay, so there's two types of inheritance. What you studied mostly probably in genetics class were genes where you change the sequence. That obviously modifies what protein you produce. And if you get a DNA sequence change, that's actually given and transferred to the somatic cells, and it's actually transferred to the germ cells, and that's going to go to the next generation. So a gene sequence change is an important way of modifying protein and cell function. Okay. But what we've more recently realized is epigenetic inheritance can also occur. Now, it used to be thought that, so you can actually you can turn a gene on and turn a gene on depending on the state of packaging. It's really clear, right? So if you package it really tightly, you can turn it off. So what's going on here is gene Y has been silenced because of the way it's been packaged in the chromatin. Okay, so there's a higher level of packaging going on that's shut off access. Has the sequence changed? No, there's been no genetic sequence that's changed. It's the package state. That's what's called epigenetics. So typically what happens is when the cell divides, that packaging state is passed on to the other somatic cell. And we used to think that when you produce germ cells, that packaging is completely erased. But it turns out that's not the case. In some cases, the state of packaging that has occurred in the somatic cells is actually passed on to the germ line and actually passed on to the next generation. What we're finding out is that a fairly quick way of affecting your progeny is by the way your DNA is packaged. And that there are studies ongoing that show, depending on what your grandfather and grandmother ate, whether their diet or whether they smoked or whether they were exposed to chemicals, affects you and your progeny. And what you're doing in your life, your whatever diet or chemical exposure that's occurring to you is going to affect not only your progeny, but your grandchildren. And so that's epigenetic inheritance. And so that's a really complicated story that's being unfolded. The big part is we can't read the epigenetic code completely to be able to follow it, but we have enough evidence to show that such things take place. It turns out that women are most vulnerable to epigenetic modifications in utero. Okay, so a woman having a child is more likely to have epigenetic changes occur to their child in utero than any other time. For men, it tends to be puberty because that's when spermatogenesis occurs. Okay, and so what you're doing at that time of life can affect not only you, but your sperm and your progeny and your grandchildren. Okay, so that's kind of a complicated story that we need to clear up. Okay, so basically where we are is we've got this DNA strip that's wrapped around the nucleosome. The way you write the code onto the histone is generally you chemically modify the tails, the N-terminal tails that are all flailing around. Okay, so proteins they have access to this part of the protein. This is fairly inaccessible, and the DNA is inaccessible. That's the rule. Okay, so how do you write the code? The code is written by covalent modifications of those N-termini. Okay, so let's take the residue lysine, okay, and it could be on any one of these. So here's the 4 different N-termini like this. So typically we use H3 as an example because it's probably the most heavily modified. And we zero in on the lysines, which are, as you know, are represented by K. So you really do need to learn those one-letter abbreviations. So this is K lysine. It carries a positive charge. Okay, and it used to be thought that these lysines, because of this positive charge, are going to help that nucleosome bind very tightly to DNA because of what's the charge on DNA? DNA is heavily charged, correct? At least the backbone. What charge is that?
Negative.
It's negative. It's phosphate, right? So if this is in the free state, at physiological pH, this is going to be positively charged, and that's going to grab onto the negative charges of the DNA. That may be the story, but that's probably an oversimplification. It may just be that there are proteins that can read that positive charge. Okay? And that is, so a free lysine means package the DNA tightly onto the nucleosome. Okay? Now, if you're going to release that, if you're going to exterminate that negative charge, you can actually acetylate it. So a histone acetylase, and we call them HATs, H-A-Ts, histone acetylate, acetyltransferase, that's where the word HAT comes from, can put an acetyl group onto that lysine, and that neutralizes the positive charge. That's a code for increasing transcription. So it loosens the nucleosome from the DNA, exposing response elements that transcription factors can bind to. So that part of the code we're pretty familiar with. Okay? So if you acetylate lysine, you neutralize the positive charge and it loosens the DNA. It promotes transcription. Okay? So when you go back to so you have HATs that acetylate the lysine, it takes, since this is a covalent modification, the only way to get rid of it is to use a new enzyme. And that's a histone deacetylase, which is called an HDAC, HDAC, HDAC. Okay, and so an HDAC will drive the reaction back to this way. Now, when lysine is in this state, it's got one of two fates. It can either be acetylated or methylated, but not both at the same time. So acetylating this prevents it from being methylated, and methylation prevents it from being acetylated. Okay, let's go in this direction now. Typically, not always, but typically methylation tends to increase the grip that the nucleosome has on DNA. It packs tighter, and what does that mean to you? What does it do to transcription? Decreases. Okay, but that's not always, it's not that simple, because it depends where the methyl group is and how many there are. You can put 1, 2, or 3 methyl groups. It doesn't change the charge, by the way. You see it's still positively charged, but you're increasing, you're putting a code on there. And a monomethyl group is different from a dimethyl, which is different from a trimethyl. Okay. So that makes it complicated. We have not decoded this yet. But one thing is true. Once you methylate this, you cannot acetylate it. You have to demethylate it to get back to lysine, and then you can acetylate it. But you can't go straight from here to there. And you can't go from here to there without going back to lysine. So you're going to have a histone methyl transfer, a methyl transferase to go this direction, and you're going to have a histone demethylase to go back that direction. To remove it, demethylase. To go forward, put a methyl on it, it's a histone methyl transferase, HMT. Okay. Okay, so that's just one. There's lots of other modifications, and we continue to learn what these mean all the time. So a serine has a hydroxyl group, and it can be phosphorylated. Okay, so we can put a phosphate group onto that serine, and that phosphate group is going to put a really strong negative charge at that position. What it means depends on what's around it. So the code is always more than one residue. It's generally more than one residue. So what this means is unclear by itself. But if the code is written properly, a protein is going to be able to recognize what that means. Okay, and so what you end up getting is you can write all kinds of codes on each one of these N-termini. And the code is, you don't write them all, but certain codes, some of these residues can only be one thing. They can only be, so this arginine can only be methylated. This one can only be, this lysine can only be methylated. But this K9 lysine can either be acetylated or methylated, depending on what the code is. This serine, number 10, can be phosphorylated. And so there's all this complicated code that's evolving. This is about 500, this epigenetic code is about 500 times larger than your genetic code that we can now sequence in about a day. We have not been able to sequence the epigenetic code yet, but we're getting there. Okay. And we haven't even talked about all the things that can happen. We've talked about this one, and this one. You can actually stick a ubiquitin molecule onto some of these, and that means something else that's part of the code. So let's look at a really simple example, and then we'll take a break. Okay, modification state. K9, when it's methylated, tends to mean that heterochromatin is forming. So your gene, you're silencing the gene. That's typically what that means. Okay. If you methylate K4, but you acetylate K9, you get gene expression. So there's a rule that's broken. If you were thinking, oh, well, all methylation means silencing, that's not the case, because it's the context where the methyl group is located. In this case, you've methylated this lysine, but the adjacent lysine has been acetylated. So that means something else. It means gene expression. Here you have phosphorylation and acetylation. Together, those mean gene expression. And these are all just on H3. And then finally, in many cases, if you methylate lysine number 27, for whatever reason, that silences these genetic genes that we'll talk about. We've already talked about the helix-turn-helix motif. Those are the Hox genes. And so you silence those developmental genes, and you inactivate the X chromosome using that histone code. Okay. So we know a few examples. We don't know by a large amount of the entire book yet. Yes?
If you methylate, does methylation of the 27th amino acid just not happen for other genes, or does it not have the same effect for other genes?
It's probably both, because you have to have both what's called the code writer, and you have to have the code reader. So both things are true. Okay, let's take about a 5-minute break there. Okay, so let's go forward now. And so part of the way of changing the way DNA interacts with the nucleosome is to modify the code. A second way is actually to insert a different isoform of each one of those 4 different histones. So I'm not going to make you memorize all of these, but one that I do want you to remember is this one example is the poster child for isoforms that can be switched out. And what does that mean? This goes back to the question that was over here. How does that affect the nucleosome function? So here's the typical histone fold. Over time, the histones have been slightly modified. The reason that they generally are not heavily mutated, what would that do to your DNA? If you had like a couple of mutations that changed the way DNA folded around the nucleosome, could you stand to have very many mutations? Absolutely not, because that would mess up all of the genes that are packaged by that method. So the histone fold is very highly conserved, all the way from a pea on up to a human and to a yeast on up. So one of the isoforms that does occur is called SYN-PA, okay? And that word SYN is actually, to help you remember what the function of this is, that tells you that this isoform is specifically constructed at the centromeric region of DNA, of chromosomes. Okay? So at the centromeric region, there's that little constriction in the mitotic chromosomes. That's also where the spindle fibers are attached. So it's a very structural part of the chromosome. And what you do is to make that centromere region function as it's supposed to be, it has to be heterochromatin. And you want that region to be heterochromatin, but you're using it not to transcribe genes, you're using it to grab on to the spindle fibers. So it's a structural part of DNA, not a transcriptional part. So to inform the DNA that is the case, you're going to turn into heterochromatin, you get rid of H3. And how would you do that? What kind of machine would you use? You want to flick out, what's the name of that type of protein? It's that big green looking thing. Chromatin remodeling complex, okay? Would get rid of that isoform and it would insert SYN-PA where that other one was. And so you're going to construct another histone octamer, but the only thing you're substituting is that one little protein for that. Okay, so it looks like a normal nucleosome, except it's got a slightly little quirk that's recognized by the DNA is, I'm going to turn you into heterochromatin and you're at the centromere now. Okay, and that has a profound, so here's the guy doing that, okay, ejecting one and putting another one in. And so it radically changes the nature of that DNA at that region. So that causes there to be constriction there. It also attracts a particular type of protein that's called the kinetochore. The kinetochore is the structure of protein that attaches to the spindle fibers. Those kinetochore proteins are recruited because those nucleosomes and the code, the histone code, is written that way at the centromeric DNA. Okay, so you zoom in, you don't need to memorize the structure of the kinetochore, but just looking at what's happening here is the DNA has been turned into heterochromatin. It's highly packaged because this is a structural element that has to be very strong, not floppy. You can't have double helixes flying all over the place. It needs to be stiff, tightly packaged, and then that's going to recruit this machine here, which is a kinetochore. So that kinetochore then, in turn, binds to the microtubule, plus end of the microtubule, and it can be used to tether the chromosomes towards the pole. Okay, but the starting point is there's two things that inform the kinetochore to form. One is that centromeric specific SYN-PA isoform that's been incorporated into the nucleosomes in this region. That helps recruit the kinetochore. And then a particular a particular lysine number 4, whatever reason, if it's dimethylated H3, that also is a signal to attract kinetochore protein. So part of it is a histone code, and part of it is a brand new isoform that's been inserted into the nucleosome. So that's how you alter the function. Okay. And that finishes up lecture 4 PowerPoint, and we need to go on to the 5th. Okay. And we're not quite to control of gene expression because we really haven't finished up the lecture 4 outline. One of the things we want to be able to do is understand the difference between code readers and code writers, so we'll deal with that. So here, this is a good example of a code reader. Oftentimes, the way they're constructed is there is a scaffolding protein to which several different proteins bind. So code readers are almost always gigantic complexes. Okay? Their job is to read the code. It's not to write the code. Okay? So the histone code has already been written in terms of methyl groups, acetyl groups, ubiquitin, phosphates, whatever, is sitting there projecting, and then this code reader has the capability of subunits that can actually read the exact combination that it's supposed to in order to cause this guy to bind. So the code reader reads it, and it will recruit other proteins. Oftentimes, what it recruits are additional code writers. So you can actually have code reader complexes recruiting code writers, and together they form code reader-writer complexes. So really what distinguishes actually a code reader from a code writer, the code reader, a writer is an enzyme. It's capable of covalently modifying the histone code. It either puts methylases on, or so removing a methylase is a covalent modification. That's writing the code. Removing something is just as important as, so adding a word to a sentence is just as important as removing a word for a sentence. So anything that modifies the covalent modifications on the histone is a code writer. Anything that binds to it and reads it is a code reader. But typically they do not have enzymatic function. Okay. So here's a possible scenario. So the DNA exposes briefly a response element, which is just a short sequence of DNA that this protein binds to. And so it sits down, and the first protein it recruits is a code writer. Okay, so this is some sort of enzyme, and let's say it puts a methyl group onto a particular lysine. Okay. Then a code reader is going to recognize that particular methyl group. Does it usually recognize just one thing? No, it's usually looking for a whole combination of different written codes. And it's going to sit down, and then it's going to recruit another code writer. That code writer puts another mark, recruits another reader, recruits another writer, and so what you get is a spreading wave of heterochromatin. So what is happening, this entire region of this chromosome is being turned off. It's being shut down. It's being turned into heterochromatin. And it can get more complicated, so the code reader can include something that reads the code, but also part of that code reading complex is a chromatin remodeling protein. Okay? And so part of the code reader is chromatin remodeling, and so it's going to it's going to grab onto the nucleosomes and pull them entire, and then that also recruits another code reader. And so oftentimes a spreading wave of chromatin will include a code reader complex that has a remodeling complex as part of its code reading. Okay. Now, how can you stop this? What if this is if it was to zip all over your entire chromosome, it would inactivate your entire chromosome. That would be bad, particularly during interphase. So there are proteins that prevent that from happening, that cause the heterochromatin to be limited to one place and one place only. So these are called barrier proteins. And barrier proteins are to prevent the spread of heterochromatin and allow the chromatin downstream from that to be transcribed. So a couple of mechanisms, there are three. The first type of barrier protein will actually bind to the DNA and bind to a nuclear pore complex and form a physical barrier that disallows these proteins to get at these nucleosomes that are part of euchromatin. So it actually physically prevents that from going forward. The second type of barrier protein does not bind to the nuclear envelope. Instead, it binds to, it recognizes DNA, and this little response element is it says, recruit a barrier so I don't turn into heterochromatin. So this protein sits down, and it binds to the chromatin that physically prevents this from interacting with those nucleosomes. Because if the code writer can't read or mark onto this nucleosome, it can't do its job. So it's blanking those nucleosomes and preventing that interaction. And finally, what can happen is a barrier protein can be have a histone acetylase component, and so it's actually acetylating these histones, which, as you know, prevents methylation. If you acetylate them, they can't be methylated. If you deacetylate them, then they can be. But so it's writing a new code that says stop here. Okay? Okay, and it turns out that HDACs and histone acetyltransferases turn out to be really complicated stories in terms of the code that they're writing, and they turn out not to be only just nuclear proteins that modify the DNA and change its transcription, but they actually modify cytoplasmic proteins. So the reason we discovered this is someone had the bright idea that making a histone deacetylase inhibitor might be a good tumor medication, because HDACs tend to pack the chromatin tighter, make it more easy to turn into mitotic chromosomes. That's what a tumor cell wants. So HDACs are very active in tumor cells. If you inactivate them, what happens is it prevents that packing, and so the tumor cell can never package its chromatin and it can never undergo cell division. So one targeted chemotherapy is actually HDAC inhibitors. And we did a problem last time doing enzyme kinetics. Now you know the basis for why HDAC inhibitors would be a good thing for tumor chemotherapy, and why tumors want to have HDACs operating. If they have lots of HDACs, they tend to package up their DNA very tightly and move it very quickly. But as I said, it gets more complicated than that, mainly because HDACs do many different things. They modify all these other proteins out here. So it's become quite a complicated story of what HDACs do and what HATs do. But we're going to leave it at our sort of simplified view is that their main function is in the nucleus to package and unpackage DNA. And specifically, both of those would be good examples of code writers. Okay. Now, let's do a clicker question here. This is also how I'm going to take attendance. So if you didn't have your clicker today, you're going to need to, is that up here somewhere? Is this it? Okay. Before you leave, you want to drop by here and sign that if you don't have your clicker.
Oh, Dr. Collie?
Yes.
What channel are you on?
It should be 41. That's the default one. Yeah. Check your clicker.
So just
What?
That's how you set it.
Oh, sure.
Oh, yeah, that's right.
Okay. Let's do that again. Polling is open. You should get like a green flash, or if you have the LCD version, it says whether you did it or not. Okay, we're gonna shut that down, so just make some determination, more for attendance than anything else. Okay. Good. All right, so what's the answer to this one, based on what we just got through talking about? This is pretty straightforward. But code reader versus a code writer. So what's the rule that makes something a code? It's usually easier to start with the code readers. I'm sorry, code writers. What's their hallmark of their characteristics? They're enzymes, and they have to make or change some mark on the nucleosome itself and on the N-terminal part of that protein. And there's only one thing that can do that covalent mark up there. I mean, there's several. Most of the things can do that covalent marking. There's only one thing that can't do it. So that's going to make what kind of mark? Phosphate group. This is going to remove a acetyl histone. This is going to demethylate it. This is going to transfer a acetyl group. So the only thing that can't do that is this guy. And that is, in fact, oftentimes a component of a code reader that's causing heterochromatin to move down the chromosome. Okay. All right. We'll go back to this. Okay, so we've really only got to this level here, the 30 nanometer solenoid. This can be fairly easily retranslated to this state. However, it's quite possible to pack it even tighter, even in interphase chromosomes. And the region that we go to next at the packing level is called the loop domain model. And so we take something like this and we pack it into loops that again make the chromatin much more tightly packed. And what happens is the proteins that the DNA are attaching to are actually matrix proteins, chromosome matrix proteins. And these proteins are able to bind to specific regions of the DNA and form these so-called loop domains. Now, this loop domain cannot in itself be transcribed. It has to actually be unpackaged, and that loop has to be unpackaged by a set of code readers and code writers to allow it to go back to the 30 nanometer and then go back to the strings on the bead. And finally, when you get to that level, then transcription factors can bind and do all sorts of fun things. But this looping out occurs for a couple of different reasons. One is it's so highly packaged that the transcription factors can't read the DNA. Okay. The second reason is it's actually going to loop into a region in the nucleoplasm that has all the machinery for forming messenger RNA. So the spliceosomes and the POL2 are not randomly distributed through the nucleoplasm. There actually is specific domains where those proteins live. Okay? So we have to go a little bit deeper in understanding the spatial domains that exist inside of the nucleus. Okay? So that's what we'll do next. First up is the chromosomes themselves. The chromosomes themselves are not randomly distributed. These chromosomes have been marked with fluorescent markers. And so this is one chromosome. This is its heterologous chromosome, homologous chromosome. And so these are the chromosomal pairs. They occupy specific regions in the nucleoplasm. We call those chromosome territories. Now, is the DNA active right here? Probably not. It probably actually has to loop into this region into a different region in order to access the transcriptional machinery. But it's always organized in a particular place. In fact, we can look at the position of all of the chromosomes. And for a particular cell type, you can actually, this is a fairly predictable location. Those chromosomes are located in a particular region. They don't just randomly occur throughout the nucleoplasm. Okay? So chromosomal territories are a way of organizing the genetic information so that it can be accessed by either the packaging machinery or the unpacking packaging machinery, and also by the transcriptional machinery. Okay? Now, to those domains. So there are neighborhoods, we call them neighborhoods, where transcription is occurring at a very high rate, and there are neighborhoods where they're turned off. Okay? So you can actually mark a gene and follow it when the gene turns on, and it will actually, there's one of those loops that's looping out, and it goes to a particular neighborhood where transcription occurs at a much higher rate. When it's in this neighborhood, which tends to be along the nuclear envelope, that tends to be where the heterochromatin is. So when it's around there, it tends to be turned off. But if it loops into a neighborhood that's transcriptionally activated, what's going to happen is the gene's going to be turned on. So you have three different genes. Gene A and C are going to loop out into this transcriptionally active gene expression area, which is called the nuclear neighborhood, and these genes will turn on. By the same token, gene B can loop out and go to a region of heterochromatin. So this will silence that gene because this neighborhood is transcriptionally silent. This one is transcriptionally active. So there's different things going on in different neighborhoods, right? And so the mobility of the DNA involves unpacking for a couple of reasons. One, you have to unpack it to get access to the DNA, and the second reason for that looping is to get it to go into the correct neighborhood. Right? Okay, so this is a mitotic chromosome, and why does it have that particular shape? That's sort of a classic shape. There's a constriction at the centromere, then you have two sister chromatids. The reason it's shaped that way is not because of the DNA, it's because of the matrix proteins to which it's attached. So the nuclear matrix protein gives the mitotic chromosome its shape. Right? During interphase, that DNA is going to collect into a chromosomal territory and is not going to be packaged onto the nuclear matrix. Could it have still one foot attached to this? Absolutely. But during interphase, it's into a nuclear territory, a chromosomal territory, and it's looping out, looping in. But the only time it actually completely is repackaged into this mitotic chromosome shape is during mitosis, and specifically prophase. That's the only time it goes back into this complete packaging state. Now, what are some examples of, let's go to the outline, examples of matrix proteins that do that job of forming the chromosome shape. So the nuclear matrix is about 1/3 each DNA, 1/3 histone proteins, and 1/3 non-histone proteins. The non-histone proteins are everything from pol 2 to transcription factors, so there's a large number of proteins in this category. These are all the structural proteins that are part of the histone code, and then there's DNA. Okay, so one of the proteins that forms the base of that matrix is DNA topoisomerase. It's a major portion of that scaffolding. Okay? And it's going to help the chromosome condense. It actually helps the sister chromatids separate, because what this protein can do is actually nick the DNA and allow strands to pass through each other and then reform them. So that's what, hence the term topoisomerase. It changes the topology of the DNA. And so that's shown in this slide here. Sorry. So you can see, here's a DNA piece. The topoisomerase, which is part of the matrix, so it's stuck somewhere in here. It's one of these proteins down there. It can take this strand and actually cut it and then pass it through, and so you can disentangle the DNA because you're going to have to pull these things apart. Okay, so topoisomerase is a good poster child for a matrix protein. And several others that we need to talk about is proteins that are going to condense the chromosome down to this level, and so we'll finish talking about those examples next time.